This is going to be an experimental analysis of our project determining optimum pH for maximum activity of trypsin enzyme. As a brief introduction, an enzyme can be defined as a molecule produced by an organism which helps to accelerate the rate of chemical reactions. They do this by binding to the substrate and lowering the activation energy required for a reaction to occur. Enzymes are essential in living organisms as they facilitate the breakdown of nutrients, making them essential to the metabolic process. Trypsin is a proteolytic enzyme that is synthesized in the pancreas of eukaryotes in its inactive form, trypsinogen. It is then converted into the active form trypsin by the small intestine. In bacterial cells, trypsin is activated outside the cell to break down molecules into smaller molecules able to penetrate the membrane. Trypsin is essential to organisms as it breaks down peptones, or soluble proteins, into smaller peptide chains. Shown in Figure 1, trypsin, with the addition of water, accomplishes this breakdown by severing peptide bond on the carbonyl side of lysine in a reaction known as hydrolysis. As trypsin is beneficial to us as organisms, it would be useful to understand the ideal environment to achieve maximum activity. For this experiment, the question of the ideal pH for maximum trypsin activity was put to the test. It was hypothesized that the activity of the trypsin enzyme would be greatest in a solution with the pH of 9. This was because the pH level in the small intestines where trypsin is active is about 9. In executing this experiment, 4 replicates were performed at each of the 5 pH levels, as well as 4 negative controls at each pH level. The negative controls were necessary as they show if there is any non-enzymatic breakdown of the substrate. Though 3 replicates is the minimum number required to identify any potential outliers, 4 replicates were executed to obtain a more accurate average and a more accurate measure of variability. The pHs tested include 7, 8, 9, 10, and 11. Microcentrifuge tubes were labeled according to the buffer that was to be added. The buffer solution for each pH being tested was obtained using a pipette aid at the end of a disposable pipette and placed in a 250 milliliter glass beaker. The reaction buffer, azocasein, and lastly trypsin were combined in appropriate microcentrifuge tubes using the following measurements. 450 microliters of the reaction buffer, 500 microliters of azocasein, and 50 microliters of trypsin. Azocasein is a milk protein with sulfonamide attached randomly. The sulfonamide is key as it absorbs light at 440 nanometers. Distilled water was added in place of trypsin, the 50 microliters, for each of the negative controls. After the trypsin was added to the first sample, a timer was started for 20 minutes. Trypsin was added to the remaining samples, and each of the microcentrifuge tubes were placed in a dry heat bath at 37 degrees Celsius after being closed and inverted several times. 37 degrees Celsius was chosen as a control in this experiment as it is the average internal temperature of the organism that trypsin is found in. Latex gloves were put on by each member prior to handling the TCA solution. After the timer went off, the samples were taken out of the dry bath, and using a micropipette, 500 microliters of a 10% TCA solution were added to each of the microcentrifuge tubes in the same order that the trypsin was added. The TCA solution was added because it stops the reaction as well as precipitating any undigested substrate. Each of the microcentrifuge tubes were then closed and inverted several times for further mixture. Each of the samples were then placed in the centrifuge and centrifuged at 12,000 RPM for 2 minutes. This step was repeated once more after inspecting the microcentrifuge tubes and noticing that a clear pellet hadn't formed. Centrifugation was necessary as it precipitates the remaining strut substrate. What is left in the supernatant is smaller peptide chains broken down by the trypsin. Distilled water was pipetted in the cuvette, and the cuvette containing the distilled water was placed in the spectrophotometer, and the spectrophotometer was zeroed at 440 nanometers. Careful not to disturb the pellet, the supernatant was then transferred into appropriately labeled cuvettes using a pipette. The absorbance of each sample was then tested using the spectrophotometer, and the results were recorded. As you can see in Figure 2, this graph depicts the average absorbance at each of the 5 pHs tested. The experimental group absorbances are shown in blue, while the negative control group absorbances are shown in red. The standard deviation is shown atop each of the bars. Important to note, there is an average 21% difference between the absorbance of the experimental and negative control groups. This tells us that the trypsin enzyme did indeed assist in the breaking down of the substrate. Additionally, there was a surprisingly less than 10% difference between the experimental groups, suggesting they all had similar amounts of enzyme activity. Finally, the standard deviation of the negative control group was 5%, much less than that of the experimental group, suggesting that there was some error in the execution of our experiment. Figure 3 shows the t-test or p-values for the experimental and the negative control groups. The left column shows the p-values of the experimental groups. The right column shows the p-values of the negative control groups. Important to note, the only p-value to be found under 0.05 within the experimental groups when compared to pH 9 was pH 11. It was hypothesized that enzyme activity would be highest at a pH of 9. The results do not support our hypothesis because there is no significant difference between the absorbance reading of any of the experimental groups. The p-value, the result of the t-test, shows the variability within each experimental group compared to the variability between the sample sets. A p-value of 0.05 or less is required in order to say the independent variable is the factor responsible for the variation. While a t-test done for pH 11 has a p-value of less than 0.05, all other t-test values are greater than 0.05 and therefore inconclusive. The choice of control variables for our experiment, after seeing the results of other groups that tested for temperature, could be improved. The PART group found the optimum temperature for maximum enzyme activity to occur is between 48 degrees Celsius and 68 degrees Celsius. For the highest possible rate of enzyme activity to be produced in our experiment, we would need to raise the temperature from 38 degrees Celsius to somewhere between 48 degrees and 68 degrees Celsius. There could have been many possible sources of variability in our experiment. Though our experimental design was adequate, the experiment was not executed properly. From an early point, it was noticed that the volume of solution in microcentrifuge tubes was too great. This resulted in an overflow in some cases, which undoubtedly changed the proportions of the chemicals within the solution. Additionally, as our negative control values were higher than the negative control values for any of the other groups, there could have been an issue with the spectrophotometer itself. Other sources of variability could have been differences in pipetting techniques, leftover supernatant after centrifugation, or slight variation in time spent in the dry bath. Between the first and second experiment, one additional replicate was added, negative controls were added, and 5 pHs were tested as compared to the 3 originally tested. To further improve upon this experiment, it would be beneficial to add additional replicates, as well as narrowing the windows of the pHs by, for example, using pH of 8.5 or pH of 9.5. As trypsin is used as a digestive enzyme by bacteria, using the results from each of the experiments conducted, the ideal condition for bacteria can be found, or for growing bacteria can be found. As outlined before, the ideal temperature for maximum enzyme activity was found to be between 48 degrees Celsius and 68 degrees Celsius. The data from the Love group, who also tested for pH, had data that proved adequate to conclusively say the optimum pH level for maximum enzyme activity would be between a pH of 7 and a pH of 9. All right, so thank you.